This is a production of Cornell University.
Okay, well, I'm excited to tell you all a little bit about my research and extension program in tomato and eggplant breeding. Okay, so first to give you just some very high level goals of our research and extension program. First and foremost, maybe the most important goal is to develop and release cultivars and germplasm that are adapted to the Northeast US with high yield, quality, and resistance to the diseases of importance in this region. We also do more fundamental work, discovering and characterizing traits that are of importance, whether to farmers or consumers in this area. I'm also really interested in modeling strategies for implementing new technologies in an applied vegetable breeding program. And specifically what I'm referring to are things like genomic selection, high throughput phenotyping. These are things that have become almost routine in some large scale breeding programs for agronomic crops. But in horticultural species, they're much more rarely used. And, there's some legitimate reasons for that. And, I'm interested in just testing out ways that we can do this practically. Lastly, I have a 40% extension appointment. And what we try to do is to generate and disseminate results on variety performance in New York for our stakeholders. So, the first thing I tried to do after I started in my position about 16 months ago was to get an idea of, who the stakeholders are in New York, what does tomato production look like, so I could focus our breeding program. And there's really quite a diversity. So tomatoes are grown in the open field, they're grown in high tunnels. We also have some of the most sophisticated greenhouse companies in the world in New York State. This is a picture of Green Empire Farms, which is a 32-acre greenhouse outside of Syracuse. Tomatoes are sold wholesale at produce auctions and to brokers and to grocery stores. They're also sold directly to consumers at roadside stands and farmers markets and through CSA shares. One thing that I found that maybe is consistent no matter where I visited is tomato diseases. But even there, there's quite a variation depending on the management practices and growing system and what specific diseases show up. So here's a look at what we focus on in the breeding program, and you can see it's a pretty extensive list. So I think maybe I have my work cut out for me. But to just kind of summarize the traits that we focus on, there are things like earliness, yield, a plant habit that's amenable to staking and easy to harvest. This is the suite of diseases that are important, whether in the open field or in protected culture in New York. Fruit quality in terms of cleanliness of fruit is very important. So that's resistance to things like cracking and blossom end rot. I'm also really interested in unique color and shape combinations. So these are tomato varieties that look like maybe some of our most favorite heirloom varieties, but have some of the disease resistance and productivity of modern varieties. And then quality of all types is important. Flavor is maybe one of the most important traits, but also one of the most difficult to evaluate. I think I might have skipped over a slide. Here it is. So here's just a snapshot of how we do our breeding. This is a picture actually of an eggplant pollination. We just did our first eggplant crosses this month, actually. Tomatoes were about a year into our breeding program. And we use a combination of marker-assisted selection and phenotypic selection. So here there's a student in the lab actually selecting the seedlings that have the disease resistance markers that we tested for. And the most important thing we do is put those seedlings out in the field and evaluate them for the traits that I mentioned. So this past summer was our first field season. We had about 2,000 early generation plants. These are all genetically distinct. And we spent a lot of time walking through and looking at these. And here's a picture of some of our favorite selections from this year. And we're excited to see how those look in the future. So I spent a lot of time talking about tomato. What about eggplant? I think eggplant's interesting because there's a bit of a discrepancy. In the United States, it's ranked 28th in terms of per capita consumption. So we eat a little bit less eggplant than we do radish and a little bit more than we eat kale. But worldwide, it's 10th in production in 2023. So there's, around the world, there's a little bit more appreciation for eggplant than there is by many of us here in the United States. Why does this matter to a breeding program based in the Northeast? Population growth in the United States is most likely going to be driven by people with recent roots in other countries. This map is showing circles that are proportional to the size of that state's population that was born outside of the United States. If you look at New York, for example, today, almost 1 in every 4 residents of New York State was born outside of the US. So I think there's a lot of potential for new market opportunities for some of these, eggplant types, and this goes for other crops as well, that are appreciated by people with roots elsewhere. So one of these eggplant types that I'm really interested in, and we've had a lot of fun looking at so far, is what broadly are referred to as African eggplant. These are 2 different species, Solanum aethiopicum and Solanum macrocarpum. These were domesticated in Africa, are still very important vegetable crops in sub-Saharan Africa and some parts of Brazil, Italy, and South and Southeast Asia. We got a small grant from the Towards Sustainability Foundation program to partner with some of our colleagues in Extension and some urban gardeners and growers across the state to do some participatory variety evaluation. We've gotten really good feedback about what people are looking for, and we're now starting crosses and hoping to have a participatory breeding program where people can make their own selections, evaluate things that have the traits that they're looking for, and still select for adaptation to this climate in New York. I want to give just a quick snapshot of one of our kind of trait discovery projects. This is one that we've been working on for about a year now. This is a project focused on salinity tolerance from a wild tomato species called Solanum sitiens. Solanum sitiens is a wild relative of tomato that is endemic to the Atacama Desert of Chile. That's a region of the planet that gets, some parts of it get about ½ an inch of rainfall a year. They've discovered populations of this species growing in what are essentially salt basins, soils with an electrical conductivity approaching 100 decisiemens per meter, which is like 40 times what domesticated tomato can tolerate. We are working with a population of introgression lines developed by a researcher at UC Davis who spent decades, moving genetic material from this species into tomato. And we're really fortunate to be able to, pick up the legacy of that research and evaluate these lines for tolerance to salinity. So this is a picture of our setup in the greenhouse. These are fertilizer injectors, but instead of injecting fertilizer into our drip irrigation lines, they're just injecting different concentrations of salt. In one greenhouse experiment, over the life cycle of the plants, we go through about a 25-pound bag of salt. And we are just looking for lines that are able to tolerate this more than a domesticated control. We're actually wrapping up the harvest of our first set of lines this week, so we're excited to see what we find. Lastly, I'll just kind of mention a little bit about our extension program. This is a picture of a field day we had last summer that I'm hoping we can make an annual field day along with some of my other colleagues in vegetable breeding and pathology here at Cornell. We invited people from, gardeners, farmers, members of seed companies to look at some of the commercial variety trials that we did. I just released a report describing one of our variety trials. It's being disseminated through different CCE newsletters. Here it is in VegEdge, so if any of you are interested, you can find it there or reach out to me and I can share that information with you. So I tried to speak pretty quickly. I hope I was within the time, but if any of you have any more questions or want to reach out, here's my contact information. And I'll thank the members of my lab and our collaborators and our sources of funding.
Is the salinity tolerance geared toward greenhouse tomato production ultimately, or?
There's kind of 2 things that we're looking at with the salinity tolerance. One is it's a big problem actually not in New York, in California, and we have some funding from the California tomato growers. But closer to here in New York, in protected culture, salinity can become an issue. It's been a challenge in high tunnels. If you exclude precipitation and apply fertilizer and irrigation water, salts accumulate in the soil. And there's ways to manage that through farming practices, but this is another angle that we're taking.
So I have a quick question. So looking into your crystal ball, right, you mentioned other crops that might be of growing importance to the state. So what would be a couple of examples of those?
That's a very good question. I don't know exactly what they would be. I'm actually part of a group that's putting together a proposal where this would be a portion of that. And we're hoping to do like a stakeholder needs assessment and have people suggest to us what are some of these crops that, they are currently growing or wish to grow, but the varieties might not be, well adapted to New York, and there's potential for breeding there.
Yeah, and then for future change in, climate, right?
Exactly. Yeah.
Okay.
Yes.
Great.
Okay. Thank you.
Thanks, Ray.
Thank you. All right, so today I want to keep it relatively short and sweet, but just kind of go over the main thrusts of the research program that I'm establishing here in plant biology. And a big focus of this is on plant evolutionary responses to climate change, where in addition to heat, really drought might in fact be one of these key drivers of rapid evolutionary change. So evolutionary change that we see not over millions of years, but of a short, small number of generations. And this is work that I've started actually from Western USA, where there was this exceptional drought that happened in California between 2010 and 2016. And so this plant, scarlet monkey flower, or Mimulus cardinalis, is found throughout California and into southern Oregon, which, as you can see, in 2010 was fairly drought-free. There was some drought in perhaps Nevada and Arizona. And so you can think of a population occurring anywhere across this range as perhaps having some amount of genetic makeup where they were perhaps more wet adapted at the time, because it was generally Mediterranean climate where you get the rains that come in the winter, and you get adaptation to having access to this flowing water in this riparian plant. But there might be some amount of genetic variation for various drought resistance genes, weedies, whatever they might be. But the severity of this drought was exceptional, and it wasn't just that any one year was particularly severe, but that year after year, essentially in some parts in the south, just water almost stopped flowing entirely in some of these riparian areas, and there was just really much more selection pressure in the center of the range as well than perhaps the system had ever experienced in terms. And so what we can think about, my lab really brings in both ecological and evolutionary aspects together. And so you can see right here, if each of these dots here is one individual, and this is their particular genetic makeup, you can see there's a crash in population. Selection's happening, lots of individuals are dying, and the individuals that have more drought adaptations are performing better and reproducing more. However, this drought then continues and continues and continues throughout here. And so there's many different selection events that happen. And ultimately, if this population is going to be able to survive, in many places, we found that evolutionary rescue was actually occurring. In other words, there was this correlation between the increase in population size and the increase in these climate-adapted SNPs, or single nucleotide polymorphisms. And so we didn't know the exact genes that were underlying this. Rather, we did whole genome sequencing and associated ultimately millions of these SNPs to particular existing climate layers, because the southern part of the range was already well drought adapted. And so we can make some inference that spatial differences in climate could ultimately help us find parts of the genome that were perhaps underlying adaptive areas. And so largely we found in a surprising number of areas that this was correlated, this sort of rebound of the populations was correlated to rapid evolution, and not just to the betterment of the environment. So really it was the underlying genetics that was a sort of this key predictive factor, which is really interesting in that essentially this demonstrates evolutionary rescue in the wild with years of demographic data as well as genomic data, with something that's fairly rare within studying evolution, contemporary evolution in natural systems. And so this is the type of approach largely that I did in my postdoc, and it's just wrapping up now, that I'm looking to kind of bring forward into my research program moving forward, where it's really looking at evolution across climatic and now phylogenetic gradients, because I want to move away from a one species framework. Essentially, this is a sample size of one, a species that we know is highly genetically diverse, and into a variety of species that might have a variety of amounts of genetic diversity. And so the targets will be genomics is sort of kind of more universal in some ways, and then we can look at the genomes, the landscape genomics of various different species, but as well morphology, life history traits that can be critical for sort of mismatches with temperature or with the amount of drought, as well as potentially herbivory and chemistry sort of work that I've done a lot in my PhD, and I'm eager to bring forward in my time here. And so we think of it so far, we have this one species where I looked at these series of populations across Western North America. But really, to get more insights, perhaps it would be useful to compare it to a closely related species that might also have perhaps certain geographic and or trait differences, and to try to understand if this other closely related species, is it also showing many of the rapid evolutionary responses that this first species is. And as you can guess, here I have a phylogeny, and so I'm going to sort of talk about these in detail, but just first focusing on this one, what we expect happens when you have two organisms that are relatively similar geographic environment. Perhaps some of the underlying population history or the traits that they experience themselves, or the type of microenvironments in which they live in these natural populations might mean that on the whole, maybe they're performing similarly because they're fairly similar in these regards, or maybe there's something kind of unique about what they're ultimately doing. So perhaps in a different environment in the prairies or out here in the East Coast, where perhaps we might see different evolutionary outcomes when you compare closely related species that are inhabiting similar environments. And so that's going to be a big thrust in my program moving forward, finding species where I'm able to essentially get a combination of different resources to try to be able to see over space and ultimately over time what's happening in these populations and how they're performing over the last few years, and then potentially deeper into time. And this is going to be potentially done through a combination of seed collection and herbarium samples. Now, I was very fortunate to have extensive collecting that was done within Mimulus. However, I can't really build a time machine, and so the best we can do is to try to find other systems where the seeds have already been collected, and I can sort of use those resources, or to start planning now for the next 5, 10, 15, 20 years in the future throughout my career to build those baseline samples. And so the strategy is going to be a little bit mixed. Currently, there are some seed collections of 50 or so species that were done around the 2010s. They're already giving some amount of time, but then also pairing these up with herbarium samples that have been they're available perhaps going on to the 1900s. And in fact, our herbarium collection here has many samples that are from particularly rich from the 1880s to 1930s. And so I've spent quite a bit of time this term sort of looking at certain target species where I have both the seeds and also potentially large swaths of herbarium samples. And sort of the proof of concept here is Black-Eyed Susan, where I have seeds available fairly deeply sampled from around 2014 across this latitudinal gradient. And then there's many, many herbarium samples just from this herbarium alone from the 1880s to 1920s. And so there's the potential then to do some ancient or near ancient DNA sequencing to ultimately come up with comparisons. And then the challenge will be, as I develop this system and potentially other systems as well, to try to get these new systems, at least new to my lab, to ultimately grow well in various greenhouse or grow chambers, or even in the field, which is something you'll appreciate in horticulture, and something that I'll be, myself and my lab will be engaging in the coming years as we seek to expand out beyond the sort of a one system framework. And with that, I'm happy to take any questions. These are the variety of different habitats in California that I've sampled for Mimulus cardinals. Yeah.
Can you infer anything about drought tolerance or morphological traits on these herbarium specimens that go back, say, 100 years or so? Is that too difficult?
It's an interesting question. So I have some of the work that I've done already with sort of morphological myself and my colleagues have done. We looked at simple things like SLA, and then I had a colleague that was looking at the actual number of cells and the thickness of the mesophyll and all these kind of more functional characteristics. I don't know. It'd be interesting to see if we could section a dried leaf and actually get any useful information out of it. It's been challenging enough to do it en masse, kind of at scale across populations within a just a living cell, really, because it's, if we're going to be quantitative, it's a lot of work. But that's an interesting idea. In terms of phenology, yeah, there's always potential to say something about flowering time, for example, in terms of when the herbarium samples were collected. The awkward bit is that you only have the bias of whoever was collecting at the time and when they were collecting. And so you're in a case where you're inferring across a variety of missing data types. But I mean, there are protocols that help sort of take uncertainty and deal with it in potentially meaningful ways. So perhaps, so I think a certain amount of it will depend on the amount of student interest I have in morphology versus genomics as I start recruiting members to my lab.
Well, thank you, Josh, and thank you, Bill, for the invite. And thank you all for the attention. My name is Vipan Kumar, and I'm Associate Professor with Soil and Crop Science. My appointment with the section is research and extension, 60% research and 40% extension. And I do work with producers, consultants, industry partners, and other stakeholders in the state, as well as in the northeastern states in terms of weed management issues. So I'm going to talk about some projects we have started. This is my second year. We wrapped up our second growing season, and I work in field crops. So I'm going to talk about some weed control projects we have started in field crops. So first one is talking about new weed species in the state, and you guys might have seen or been heard about Palmer amaranth or pigweeds. There are two new species have arrived in the state, Palmer amaranth and water hemp. The first two pictures you see, those are Palmer amaranth inflorescence. One is a male, one is a female. The second picture is Mike Stainyard, our Cornell Cooperative Extension team lead for Western New York. He was scouting some fields this growing season and asked me to come, and we found some new reports of Palmer amaranth in Yates and Ontario County. These are this growing season from soybean and corn and alfalfa fields. We had like 6 different grower fields where we saw Palmer amaranth. So these two pictures on my side is the water hemp, another pigweed, highly invasive, highly problematic. Both pigweeds are basically very, very problematic in Midwest and other southeastern states, but also has arrived now in the New York. So what we are trying to do is we are characterizing these populations for any kind of herbicide resistance evolutions. We are doing greenhouse testing. We are doing some field experiments, looking at different herbicides. An example here in this slide is we are doing some dose response studies looking at glyphosate resistance. And if you see, there is a New York population and Nebraska population side-by-side comparison for water hemp showing resistance to Durango, which is a glyphosate active commonly used in our field crop systems. The picture on my side is my graduate student, Enrique Scatina, is a master's student under myself and Dr. Lynn Sosnowski, holding a water hemp plant. This is a glyphosate-resistant water hemp plant near Waterloo, growing in soybean. He's looking at some options, herbicide option that we can use, alternate options, pre and post herbicide options, as well as looking at the growth as well as reproductive characteristics of some of these herbicide-resistant population. Along with those, water hemp and Palmer amaranth, we have also been testing some grass species like annual ryegrass. These are some population came from grower fields who are using annual ryegrass as a cover crop. And those cover crops, they were not able to terminate with glyphosate. So they have becoming a problem now as a weed. We are testing those. Along with those species, we are also investigating other problematic species like lamb squatter, common ragweed, giant ragweed, some other grasses like foxtail. So this is a kind of long-term ongoing projects got funded by Hatch and Smith Lever funds this year. And we are trying to look at as a regional approach from other states as well, multi-state project, characterizing resistance and locating those resistance population in the Northeast. Another example is a horseweed population. This is a very typical response we are seeing in horseweed population from New York. This is a population we collected, actually been sent by our CCE folks from Central New York. And we've been testing this with different herbicides, again, commonly used herbicides in soybean and corn. We are seeing issues with the Roundup, as you see here on the picture, compared to the non-treated. We have soybean products like Cobra, Synchrony, even corn products like Callisto showing issues with controlling these horseweed population. Very consistent response. We have 10 populations screened so far. Similar response we have been seeing. These are again the pictures of annual ryegrass just sent by the growers. You see the picture on the very left from last year. This is the same field in this growing season where he had wheat, and he's been dealing with this glyphosate-resistant annual ryegrass. We are trying to figure out what alternate options, alternate methods we can employ, and we have put together a AFRI grant just to look at the wheat seed bank management in wheat soybean rotations, what that producer can use. And the middle picture is again another annual ryegrass cover crop was not getting terminated with the 2 shots of Roundup in Ontario County. So you can see very quickly some of these cover crops can also be a problematic as a weeds. So along with those herbicide resistance screening and testing and characterizing, we are also looking at field works, at especially product evaluation program, looking at alternate option. As I said, we want to give producers the immediate options they can use to control these herbicide-resistant weed populations. These are pictures we have one of my graduate students looking at options, herbicide options in different trait technologies like conventional corn or Roundup Ready corn. What options do we have to control glyphosate-resistant water hemp? This is a picture from Wayne County. Producers growing conventional corn, a silage corn. What options do we have to control this glyphosate-resistant water hemp? Similarly, these are producers' on-farm studies. We are looking at options like a pre-applied as well as a post-applied herbicide options for controlling glyphosate-resistant water hemp as well as other weed species. In addition to product evaluation or herbicide-based weed control, we are also looking at non-chemical weed control tactics. This is my graduate student, PhD student, looking at side-by-side comparisons on innovative weed control tactics such as interrow mowers, electric weeder, as well as harvest weed seed control. How those tactics we can integrate into our system as a holistic approach for targeting those weed seed bank management. So this is again another way of looking things from the system standpoint. We are integrating some of our precision ag tools. One of my PhD student also looking at precision ag tools such as the sensors and cameras, how we can use the drone-based imagery to detect and identify weed species in the fields, and that information can be used to make decisions for controlling those weeds, because artificial intelligence is quite popular now, and we want to use that technology to identify and manage these weeds more site-specifically and species-specifically. Looking at these, integrating these chemical, non-chemical weed control strategies, as I said, some of these projects being funded by USDA and NIFA, looking at multi-tactic approach, including cultural practices, including precision ag, including mechanical approaches like harvest weed seed control. Overall, I guess my overall vision for the research and extension program is to develop sustainable and economical IWM strategies for the producers. As I mentioned, my program looking at different aspects looking at the cultural, chemical, precision ag, and mechanical tactics, how we can integrate them together to help producers to make those decisions how to control these tough to control weed species. I have not put together any slide for my funding agencies, but there's a lot of support from New York corn soybean producers, industry partners, as well as USDA and NIFA. Been working closely with some multi-institutional teams like a GROW team, which is called Getting Rid of Weeds, run by USDA ARS, Dr. Stephen Mursky at Maryland. So we are educating producers on some of these issues and helping them to make some of those decisions how to control these tough to control weeds. That's my last slide. Thank you. Yes.
I'm wondering about this for the second time, so I'll ask you now. Your picture of I think it was the horseweed control, it actually looks like the glyphosate treatment is more vigorous than the untreated control. Is it conceivable that the resistant population somehow grows even better in the presence of glyphosate?
Excellent question.
It's a visual thing. So
Yeah, absolutely. We haven't done any work to look at closely, but there are examples in the literature that shows that because of resistance mechanisms, we don't know what mechanism we have. There are examples in GMO rice that has shown with mutations, with the increase in copy number, EPSPS gene copy number, that is the mechanism of resistance to glyphosate. In those GMO rice, they have found increase in yield. They have found increase in growth. So that is called fitness. We will be doing some fitness studies to look at the fitness cost, if there is a cost or there's no cost or there is a benefit of that fitness. Some of those things help us to devise strategies how we can implement our weed control tactics. Definitely excellent question. And being from weed science background, we have done some of that work in kochia where we did not find any fitness cost due to glyphosate resistance. But again, these population we are dealing with in these water hemp, palmer, or horseweed, they are probably not only resistant to glyphosate, but multiple resistance to other chemistries. So especially resistance to oxenics like 2,4-D dicamba, we have seen there's a fitness cost. So it's a matter of what resistance we are talking and what mechanism we are talking that can determine the fitness. Any other question?
I have one question.
Sure.
So the plant biology lightning series on Friday, Ginny Moore from Plant Breeding Genetics gave a pitch on her program around breeding for sustainable cover crops, right? But you're talking about the problem that they're becoming too sustainable, right? So they're weedy. So in terms of adoption of cover crops, how much is weediness a really huge concern?
So this plant, the annual ryegrass, I'm talking in general is tough to control, tough to terminate if you're using a cover crop, and there is a specific guidelines if you're going to use a chemical to terminate these cover crops. What we are trying to educate producers, if you're going to use this as a cover crop, be proactive when you're going to terminate, make a better plans, what weather conditions you're going to have when you're going to apply those glyphosate to terminate it. In general, irrespective of resistance, this plant is hard to control. And we are trying to educate, like this is a closely related species to Italian ryegrass. Producers in the South, they are dealing with this more aggressively because they were using annual ryegrass as a cover crop. And the problem there is some of these seed companies are selling the annual ryegrass as a VNS, variety not specified. There are mixtures coming from the South. Some of those seed bags are having, varieties from different regions, and that cause problem. I don't know, I have exact answer how we can, increase the adoption, but there are species which are perfectly fit in terms of, if you're going to use chemical to terminate those. But there are other methods to terminate those cover crops. Definitely cover crops are foundations for suppressions, weed suppression. Folks in the New York State are pretty excited about other benefits, but I think weed suppression is, again, top top benefit producers in the Midwest and other states, they are looking for and adopting cover crops, especially cedar rye, if we talk about cedar rye. So yeah, I guess depending on the goals of adopting cover crops or growing cover crops, it depends how you're going to terminate, and that's what we are trying to educate, plan properly, what you're growing, for what you're growing, and how you're going to handle this.
Okay,
Yeah.
great.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.